Thank you for participating in the Mandalis fiscal year end 2025 business and financial report result briefing. Here I am making a forward-looking statement as usual, then going to the presentation. So before getting to the main topics, for those new to Mandalis and also for coming back after the considerable period, I'm going to briefly touch on what we do and who we are. So Modalis is the first epigenetic editing company founded in 2016. So we have 10 years history right now. And we are the first group that is established this technology with using CRISPR technology, which is originally discovered as gene editing, but we engineered it to make it as an epigenetic editing technology. And with that technology, we have demonstrated a very robust efficacy without serious toxicity. And then we have established pipelines primarily for muscular dystrophies, but also for CNS indications and also cardiovascular. So it has an unlimited opportunity, upside of opportunity for this technology. One of the challenging point for the gene therapy is manufacturing, but we have established a manufacturing process for this engineered capsid technology and with reasonable productivity and also quality as well. And those development has been supported by experienced team. And also I wanted to note that our technology and also programs are supported by a strong IP portfolio. This slide shows how our technology works. The technology is named CRISPR guide nucleotide directed modulation, or in short, CRISPR Gundam technology. So it consists of three components. One is guide RNA as a pointer, and also Cas9 as a binder to the DNA. And the thirdly, which is most important, is the epigenome editor. So epigenome editor work for activating a gene or silencing a gene. So we have two different components for different purpose. So using this, we are able to activate a specific gene for a genetic disorder caused by excessive amount of gene expression. We silence the gene. And for the genetic disorders, which is caused by lack of or less amount of expression, we activate the gene to treat the genetic disorders. So to deliver this molecule, we use a virus vector. We more specifically AB vector. So we code that Gundam molecule in the DNA form and pack into single AB vector. So the vector go into the target tissue and decoded as protein and expressed as a protein and bind to the regulatory element of the target gene, so that we can upregulate or downregulate the target gene specifically, which leads to efficacy. So our strength is primarily in the payload. So it's really important to get access to a good delivery technology. And we have established a multiple collaboration with multiple group, including JCR Pharma, which has a very strong vector technology, and also Genix Care, a Korean company, and also Broad Institute. So combining our Gundam payload with those the muscle tissue tropic capsid, we are able to develop a very powerful gene therapy technology. So, and the benefit of that is three things. One is it just need single injection, and that leads to a long-lasting effect. And thirdly, and most importantly, unlike the steroid, which just provide the care, Gundam provide the cure, disease modifying cure. When we started the development of with epigenome editing technology, we are the only company doing so. However, after that, there are a number of companies started to develop with similar technology, as listed here. But still, we are uniquely differentiated from the other company because unlike the other company, which primarily focus on silencing the gene, we focus on the activating gene in the belief that the silencing can be done with multiple technologies such as siRNA or antisense oligo. However, activation is difficult to target. So if the gene size is within a reasonable limit, that can be the primary choice is the standard gene therapy, gene replacement therapy. However, for those indications with large size gene, large size disease causing gene, this approach is the only approach that can treat the genetic disorder. Okay, with following slides, I wanted to touch on the topics listed here, starting with the business highlight of fourth quarter 2025, followed by financial update, and then growth strategy and the summary at the end, Q&A we got through our website. Okay, for the first part, business highlight of the first quarter 2025. As listed here, we have four topics here. First topic is a review of MDL-101 progress and the timeline, and the secondary progress in MDL-103 for FSHD disease. And the thirdly, progress in IP, and lastly, finance and the others. Okay, starting from the first topic, but before going into that, this is our current pipeline. So the pipeline indicated by the blue is the muscle disorder, the pipeline for muscular dystrophy, and also green one is for cardiomyopathy. And also a yellow three pipeline are for CNS indication. Okay, the target for our lead program in MDL-101 is LAMA2 congenital muscular dystrophy, or also known as CMD type 1A. This is a very severe disorder caused by a mutation in the gene called the LAMA2. And the prevalence of the disorder is 8.3 in 1 million. This is a very rare disorder. However, in United States, it is estimated that the 2,500 patients are in the United States. So we are making progress toward our fast in-human trial. So in this slide, I'm going to give an update for that. So that's 4 main points to enable the clinical trial. The first one is a GLP tox study with non-human primate, and the toxicity study are ongoing, and additional analysis are being conducted to enhance the certainty of the clinical transition. That's the first point. The second point, mouse IND enabling. The study is in progress smoothly, and the interim analysis demonstrated significant survival benefit and also functional recovery compared to control group in the disease model. And with no issue expected for completion, so this is going very well. So this data will be presented in coming conference such as SGCT in May. So we will have opportunity to share more detail there. The thirdly, manufacturing and the investigational drug AB manufacturing or GMP manufacturing is underway. And lastly, for clinical preparation, we have completed interviews with U.S. clinical candidate sites and the PIs, primary investigators, and we are selection are in progress. For clinical trial outside of the United States, as a backup plan, a term sheet agreement has been signed with a pharmaceutical company in that relevant region regarding the conduct of the clinical trial as well as the future commercialization within that region. So it's we believe this is a validation of our technology and a program. And so we are very happy to announce this. And also preparation for barrier separation study or testing and development are underway. So before touching on the adjustment, we announced on the sideway of our fourth quarter report on February 12th. I wanted to touch on our MDL-101 development status and tell you what has changed and what hasn't not changed. So let me start with the unchanged foundation. So first of all, the cryo survival extension has been confirmed in our mice IND enabling study. So there's no change of mechanism of action or therapeutic concept. It's still robust. And there's no safety concern in that mice study as well as the GLP monkey GLP tox study. So there's no safety concern coming up. So therefore, I can tell you that there's no impact on the platform technology. And also, as I mentioned in the earlier slide, we have established external validation supported by the term sheet rebel agreement with our regional company. So toward the clinical development, there are four main steps. Started with a concept validation, followed by non-clinical development, and then clinical transition, which we are in right now, and then clinical execution. So here we are implementing the strategy development optimization with the following reason. First, we think there need to have a further optimization of the clinical transition preparation, and also refinement of the development parameter is required. And the thirdly, enhancement of translational reproducibility needs to be made. So that leads to IND timing reassessment. But these all are for increased probability of clinical success. So as a result, we have a higher chance of the success and a better and even shorter time to reach the clinical proof of concept. So we view the current development stage as a natural evolution toward the clinical execution, reinforcing the long-term development trajectory. So with this slide, I wanted to touch on the key points of development optimization I just mentioned toward the clinical transition. So this is to confirm biological validity and promote optimization for the clinical transition. First point is that the biological foundation maintained as is and even strengthened. Because we have a very clear survival extension compared to the control group, as well as the functional improvement. So mechanism of action and the pharmacological efficacy consistency supported with the mice study I mentioned earlier. And also, this mice study and the NHP study supported that no new safety concern lies at this time. So there's no change to the treatment concept itself. So then here we are introducing refinement of the development for clinical transition. The points are conduct additional analysis to enhance the certainty of transition to clinical phase and strengthen translational reproducibility through refinement of development parameters. And lastly, this is to maximize the success probability through the data-driven decision making. So then we have a clear next steps. First, we're going to complete additional analysis and optimize the development plan. And then after doing so, we ensure the alignment with the regulatory authority. In the case of our development, FDA, and then so then this leads to reviewing IND filing timing to ensure the optimal clinical readiness. And also, meanwhile, we conduct the site preparation in parallel. So this gonna lead to the coming trial. So overall, by prioritizing the optimization of biological and the clinical readiness, we aim to maximize the probability and the long-term clinical success. That's the whole point of this alignment. So so in the last December, we have announced the clinical. We have shared the clinical data with our publication on the human gene therapy. This highlights the clinical data we have conducted mice study and also monkey study. So we are able to demonstrate the reasonable RAMA1 upregulation that compensates the RAMA2 disease-causing gene to the level that we can expect the clinical benefit. So if you are interested in, you have an opportunity to check this out on the website because this is in the public domain. The other things I wanted to touch on here is that we have reviewed the trial design of the clinical, and we have adjusted the number of the patients recruited for this clinical trial. And we actually downsize it to 3 patients for low dose and 3 patients for high dose because we are able to see robust efficacy in our mice model. So we believe that 3 is the enough number to see the benefit in this trial. So this will lead to the shortened timeline in the clinical trial. So we believe that we are able to reach the clinical proof of concept even earlier, even we are introducing alignment in the clinical stage right now. Okay, then switching gear, and I wanted to touch on our MDL-103 program that is targeting for FSHD. So this is a larger number. This disorder has a larger number of patients, and then this disorder is caused by a gene mutation called the DAX4. So the product of this DAX4 coded gene is toxic to the muscle, and that leads to the muscular dystrophy type of phenotype. So I don't go into the detail of this disease mechanism, but so the DAX4 gene is coded on the chromosome 4, and this is silenced in the healthy people, but due to some reason, the silencing regulation is loosened, and then the DAX4 coded gene are expressed, and that leads to the problem. So in the last quarter, we announced the result of our disease model study. However, for this quarter, we added more encouraging data. So the disease model we are conducting is as highlighted on the left-hand side of the slide. So in short, we inject the patient-derived muscle cell into the mice so that the patient muscle cell are developed in the mice body. So then we injected our molecule to see how much we can suppress the DAX4 gene. However, DAX4 gene is rarely expressed in the muscle. It's hard to detect. But we are able to see that by checking the downstream gene, the three downstream gene listed here. As shown on the right-hand side, in the middle top, we demonstrated the strong suppression in the local injection system in the last quarter. As you can see here, the disease model mice treated with the Ganda molecule have very strong suppression of the downstream genes of DAX4. But this time, we are sharing that's even stronger in the systemic injection model. So the same molecule injected systemically for this time, and we are still able to see that even stronger suppression of those DAX4 downstream genes. So this is very encouraging data, and we believe this is the preclinical proof of concept, and with the human cell. So in this case, we are able to use the human construct, not the mice surrogate construct. Human construct demonstrated this efficacy. So this is a very encouraging data, and we are convinced that we are able to move forward to the development stage. So this study is supported by a grant from the Solve FSHD and the X Prize. So we are talking to those grant order for the future development at this point. So this data result will be presented most likely at the ASGCT meeting in May. So you might have opportunity to see more data in the coming conference. Okay, so with those progress, here are the coming milestones. As I mentioned earlier, we are in the review of the MDO-101 timeline. So we will share the more detail once we have a clear idea on the timing of the IND filing and also the other milestones. And for MDO-103, as I said, we have established a very strong animal proof of concept. And on top of that, we are talking to multiple pharmaceutical companies for the collaboration. So we are expecting the business development milestones this year too. So this is a summary of what we have achieved and what are the coming milestones. Most importantly, the MDO-101 timeline is in review, but we are sharing here that we are targeting clinical proof of concept establishment 2027 to 28, and the fighting for the conditional approval probably later 2028. And also for the other program, we have established very strong preclinical proof of concept for MDO-103, including those we are making very robust progress, and also making business discussion with multiple parties. Very much. So for IP side, we also have made the progress, and the 3 patents have been granted in Japan in 2025. And one is for MDO-101, our lead program, and the other 2 are for MDO-103 for FSHD and 104 for tauopathy, including Alzheimer's disorder. So you are very happy to see those progress because this strengthens our IP portfolio as well. So in the next section, I'm going to touch on the financial update at the end of 2025. In 2025, at the end of 2025, we have the cash of 2,800 million, about 208 million yen, or in US dollar, about 19 million US dollar in the bank account. So this is a slight decrease from the at the end of 2024 because of the development activity. But we still have a good amount of cash that support our development activity, mainly for MDO-101. And then on the PL side, we have spent about 2 billion yen, 2.2 billion yen last year, primarily for the development. And this is about two times more than the last year, 2024, because that the development are moving towards the clinic, and we have more activity in the on the R&D side. And in our US dollar, we have spent about 13 million US dollar for R&D, and including that, we have spent about 15 million US dollar last year. Okay, so then the next topic is the growth strategy, which hasn't been changed. So as mentioned in the previous slides, we are focused primarily on MDO-101 development, which is the crown jewel of our pipeline. But also that progress opened up the opportunity for larger indications such as MDO-201 for DMD, 202 for DM1, and also 103 for FSHD. So those share the same platform being developed for MDO-101. So once the clinical proof of concept is established for 101, this will strengthen the technology position of the other muscle disorder, as well as the other pipeline for a CNS indication as well. So MDO-101 is a relatively small indication, but this is untapped opportunity, and there are no material development besides us. So this is a completely blue ocean, and because our technology is the only technology that can address the disease mechanism. So this is a very unique opportunity for us. But the same concept, same technology is shared with for MDO-201, so we can extrapolate the result for the indication like that. So this part is summary. So key takeaway of MDO 2025 is, first of all, the preparation of MDO-101 clinical trial application have been progress to the clinical transition phase. And we are currently implementing a development optimization, which will take a little time, but the transition is currently underway. The secondary, we are sharing the outlook for the clinical trial plan and set the target of clinical proof of concept 2027 to 2028 and the conditional approval application in the later of 2028. And also, we are delighted to share that the progress of MDO-103 too, and we have demonstrated robust efficacy in the disease model. And we believe that is establishment of a preclinical proof of concept. Okay, so with that, I wanted to close the presentation part, but I wanted to touch on the Q&As that we got through our website. So the first question is that is there a problem with the treatment concept of MDL 101 or Gundam itself? Because we are saying that we are implementing some adjustment. But I can tell you that the concept is strongly holds, and because in the IND enabling study using the disease model mice, we are demonstrated critical survival extension and also functional benefit. So we are very sure that the concept holds, and we recognize this and a matter of pertains to the development optimization for the clinical transition, not the therapeutic concept itself. The second question was, are there any safety concern? And I can tell you that no, at this time, no new safety concern have been identified, and the current effort are primarily aimed at enhancing development certainty. The third question is, why is it necessary to review the timeline now? The answer is that we prioritize biological and the clinical readiness at the transition point to the clinical phase over adherence to the fixed schedule. So we believe it is crucial to maximize the probability of the long-term success through the data-driven decision making. The fourth question is, what impact will this have on the other pipeline and the platform? I can tell that it's none. This is uniquely to MDO101 and no effect on the other pipeline or the platform as well. The fifth question is, what are the next specific milestones? And for MDO101, the following the completion of additional analysis we are conducting right now, the confirmation of regulatory alignment is followed, and then finalization of the optimized development plan. So then we will proceed to the preparation of IND application. So that's the coming milestone. And also for the other program, we are expecting a business development milestone. So 2026 will be a fruitful year for Modalis. And then lastly, the question number 6 is, does this not suggest an essential challenge regarding the translation into human languages? And the answer is that the data obtained to date consistently support the mechanism of action and the pharmacological activity. So, including a clear survival extension in the disease mice model and also functional benefit. So, the current initiative aims are not at biological validity itself, but at optimizing development parameters and enhancing translational reproducibility at the transition point to the clinical phase. So the company Modalis maintains its recognition of a therapeutic concept validity. So we don't see that this is this does not suggest essential challenge to the concept. Okay, with that, I wanted to complete the Q&A section as well as the whole presentation for the fourth quarter 2025. So lastly, I wanted to say thank you and stay tuned with Modalis.